Hi, today you'll be learning how to design 2D and 3D DNA origami structures utilizing the already pre-built in 2D and 3D shapes that are included with the ATHENA software. The plan for today's video is to first go over a basic overview of ATHENA, then we will design 2 helix bundle and 6 helix bundle 2D structures, then we will design 2 helix bundle and 6 helix bundle 3D structures, and finally we will visualize the structures that we just designed using Chimera. When you first open up ATHENA, this is what you should see. The left side of the interface is generally used for more artistic purposes, such as changing colors or displays, whereas the right side of the interface is used for the actual design of the structure. Therefore, for the purposes of today's video, we will be focusing on the functions that are included on the right side. Now let's design a 2D structure. Up here, we can see that there are already 2D examples, so we can just open that up and choose one. For example, the hexagon. Then we will keep our scaffold as default. And down here is where we will choose based on if we need to design a 2 helix bundle structure or a 6 helix bundle structure. Using Paradix will give you a 2 helix bundle structure, but using Metis will give you a 6 helix bundle structure. Note that you can also change the minimum edge length, but that really depends on how big you want your structure to be. For now, we'll just keep it as 42. Then we will click Run Sequence Design. And once that has completed, we will save our structure. Make sure to include the PDB file, as that will allow you to visualize your structure in Chimera. So we'll click Save. And I will save it to my desktop, but you can save it to wherever you want. And you can see that it has saved right there. Designing a 3D structure is very similar, but instead of using 2D examples, you'll be using 3D examples. I will keep my scaffold as default, and down here is where you can once again choose whether to design a 2 helix bundle structure or a 6 helix bundle structure. If you use Daedalus, you will get a 2 helix bundle structure, but if you use Talus, you will get a 6 helix bundle structure. You can then click Run Sequence Design, and once that has completed, you can save your structure accordingly. I have now opened up the folder for the 2D 2 helix bundle hexagon that we created earlier in this video. And as you can see, there are many different components in this folder. But some components to notice are the BILD and PDB files, because you can use those to visualize your structure in Chimera. I'll be demonstrating that in a second. Another thing you should note is the CSV file, because that contains the DNA sequence for the scaffold and staples, which you'll need to order in order to actually experiment with your structure in the lab. I have now opened up one of the PDB files in Chimera, and as you can see, our structure is displayed right there. We can use Chimera to move our structure around to visualize it. We can also take different actions such as changing the color, we can change different labels, we can also look at different chains and residues. For now, that's all we will be going over in terms of Chimera, but thank you so much for watching the video, and I hope it was helpful in guiding the design of 2D and 3D DNA origami structures utilizing ATHENA.